All right, let's talk neural crest migration. This is a cubist neural crest image. Looks like stained glass to me. It's very abstract. All right, let's move on. Okay, so we're going to talk about, so when we talked about the formation of the neural tube, the neural crest was that region between on top of the neural tube, which splits off from the epidermis to allow it to be separate and not connected to the epidermis. So here we have this image of, this would be the neural crest up here in the blue. And they undergo epithelial to mesenchymal transition. So when the skin hold on a second. So it's because I'm on the wrong thing here. All right, let's go to the next picture. Okay, so here's the image of, or kind of the picture of what's going on that we're talking about. So previously we talked about how the notochord kind of instructs these cells to become the neural plate. Then we have this folding where the hinge point starts to come together. We have the forming of the neural tube, and then these layers of epidermis, the kind of dark blue that come together. But then in between where this hinge point was, we have the neural crest, right? And so those cells have to kind of get up out of there in order to for this tube formation to be done, right? So you can see here that there's room between the neural tube. The neural tube is not connected to the epidermis, right? Or the notochord. They're all independent. So we start, so I'm going to kind of go over these steps again real quick. So we have the apex of the neural folds up here. That is what will become the neural crest. Their delamination will occur. So these this delamination occurs at the point of the neural tube actually closing. So you can see here we have the neural tube finally closing, and we get the delamination occurring. And then they migrate out of the ectoderm tissue at that point, and they will go on to become other things. So what will they become? So these neural crests will migrate and go on to do other jobs at that point. These are some of the things that these migrating cells will turn into. So they'll turn into the peripheral nervous system. So we have neurons, including the ganglia, Schwann cells and other glial cells, things we talked about in the previous lecture. They'll become endocrine or paraendocrine derivatives, so adrenal cells. They will become pigment cells, so melanocytes, etc., so epidermal pigment cells, facial cartilage and bone cells, as well as some types of connective tissues. So here's kind of an image depicting in a chick the regions of the neural crest and kind of their migration, going to subsequent migration. So the cranial neural crest migrates into the pharyngeal arches in the face to form the bones and cartilage of the face and neck. So you can see the pharyngeal arches right here. So these cells up here will migrate forward out of the neural crest, and like I said, become the face and the cartilage of the face and neck, and also the cranial nerves in your head. The vagal neural crest, which is these top ones right here, and the sacral neural crest, which are sacral crests down here, form the parasympathetic nerves of the gut. So they go and interact with your gut and control your gut action, so to speak. Let's see, where am I here? The cardiac neural crest arises near somates 1 through 3 at the very top here. So you can see that. They're critical for making the division between the aorta and the pulmonary artery. The neural crest cells on the trunk. So these down here. They make sympathetic neurons in pigment cells, so the melanocytes, and a subset of them at the bottom here, subset somate 18 through 24, form the adrenal gland, or the medulla portion of the adrenal gland. So you can see as these cells migrate out, they go and they do have a lot of other functions. So we had facial tissue, we had the control of the gastrointestinal tract, we had the cardiac crest, so that was the division between the aorta and the pulmonary artery, etc. So they have a lot of function and a lot of stuff to do after they are done or formed and get out of that, form that gap there. So this is kind of just an image showing lineage tracing of some of the trunk neural crest cells in a mouse. And so they, before migration, so pre-migratory non-neural crest cells were marked with with fluorescent tags. So they call this a confetti mouse, as they could introduce up to 10 different colors to these different cells. So they did 10 different fluorescent tags. And so they could then watch where these cells all migrated to in the different regions and where they ended up. So in part B here, they followed those individual colors to their faded structures later in development. Fluorescent cells were seen in the dorsal lateral pathway where melanocytes differentiate. So remember, those are the pigment cells. In the dorsal root ganglia, the part of the Schwann cell population and the sympathetic ganglia, so that's these ones, migrated to. So you can see that these dorsal neural tubes. So essentially what they're doing, I guess kind of a 1,000-yard view of what they did is they multicolor stained these different neural crest cells, right? So they had 10 different tags, as you can see here, all these different colors, fluorescent colors coming up. And then they went back and at day 10.5, so we started with day 9, so this is only a day and a half later. But remember, mice have a very much faster development, and it's not 9-month gestation like it is for humans. Anyways, so a day and a half later, they went and they looked and at all the different tissues and saw what cells were stained. So originally, those cells all arose from this neural crest. So those are the only ones stained. So the only ones on day 10.5 were ones that originally were in that location. So they kind of did a fate mapping, right? And so they found Schwann cells, sympathetic ganglia, dorsal root ganglia, melanocytes out here towards the epidermis. So those are going to be obviously where your pigment is, towards your skin, Schwann cells and other glia, etc. So a much more succinct way of saying it is that these, they tagged them and they kind of saw where they went, and they ended up becoming a whole bunch of different types of tissue. Let's make sure I didn't skip anything. So here we kind of, they've kind of not only done fate mapping, but some of the established some of the genes that are differentially expressed to cause these different specifications of these cells. So here we have these neural crest progenitor cells. So those are the ones that are migrating. They haven't really become their final form yet. And they we don't have the gene for this, but they differentiate into these two different types. These can be either cartilage, glial, or neurons. This subset can either be glial, neurons, or melanocytes. They'll differentiate into these neural crest lineage precursor cells here. And then if this cell expresses EDN1, it will become cartilage and bone. If it expresses neuroregulin, it will become a Schwann cell or a glia cell. Similarly, this precursor that can either be a Schwann cell or a neuron, if it expresses neuroregulin, it can go this way. So you can see there's kind of multiple paths here that a cell can take from that neural crest, where can start as a GMN or a glial neuron melanocyte, and then end up as a Schwann cell, or it can end up as a melanocyte, depending on what is expressed. If EDN3 here. So what's interesting here is that something like Schwann cells, neuroregulin is important to get it to become a Schwann cell precursor. But in this case, in this cell, only EDN3 needs to be expressed to get it to this precursor stage. Then both of them need to be expressed for them to become Schwann cells in the end, or glial cells in the end. But and the 1,000-year view for this one, kind of the whole message is we have these stem cell-esque progenitor cells, right, where they have 4 different fates, and they've all migrated from the neural crest. Depending on what genes are expressed, they differentiate into these 4 different cell types, right? Okay. So specification of the neural crest cells. So the neural plate is bordered by neural crest anteriorly and caudally, and by placoidal ectoderm anteriorly. So remember, anterior, posterior, anterior towards the top, posterior towards your bottom. If the ectoderm cells receive both BMP and WNT for an extended period of time, they become epidermis, right? So that will help create that epidermal layer up there. If WNT induces BMP, then we get placoidal cells. If WNT induces BMP but also remains active, so you can see this is kind of time this way. So in the case where both WNT and BMP are present the whole time, we get epidermis cells. In a case where we have WNT that induces BMP and then WNT stops, they'll become placoidal cells. If we have a situation where WNT is expressed for a while and then it expresses BMP but still keeps going, so at the end you started with WNT but then you end up with both, then you get a neural crest. And then in the case of the neural cells, which you can't read back here, it says neural cells, it's where only WNT is expressed. So that's kind of how these this neural crest specifies. So it gets even more complex. We're not going to go into this too much, but that's what I'm going to show you. And this is kind of simplified here, but you can see all these crazy interactions in the neural ectoderm and the non-neural ectoderm. Simplified, you can kind of see this pathway where WNT, BNT expresses MSX1, which self-expresses MSX1, which expresses FOXD3, etc. So there's a lot going on in this pathway. I just want to show how complex this circuitry is of gene expression in cells from the early ectoderm and this migration. So they kind of tried to give you an analogy of neural crest and the migration here, where we have these pathfinding exons that kind of go out ahead of time, and then the traffic here is all the cars are the migrating neural cells. The pavement is the substrate that they're migrating across. We have more individually migrating neural crest cells up here, and then we have these local guidance cues or environmental, they're biological factors, but factors that help to guide them to their prospective zones and what they're going to become. So the traffic cop here would be those gene expression, differential expressed genes that lead them to become glial cells or whatever. It's not the greatest. This was with the textbook. It's not the greatest analogy. Okay. So let's talk a little bit more about the neural crest delamination and migration. So the process of neural crest delamination is shown here at the time when the neural and surface ectodermal cells, ectoderm, have separated and are both in the process of fusing at the midline to become the neural tube and the epidermis respectively. So this is right at the point of fusing, right? BMP and WNT signals specify the three major regions of the neural epithelium. So we have, where's BMP and WNT pictured on here? BMP and WNT. Okay, so those are specifying the three major regions of the neural epithelium up here, which are distinguished by their expression of unique adhesion proteins. So remember, what's causing them to not stick together or stick to only their specific thing are these cadherins, right? So you can see here we have cadherin 6B, N-cadherin, and Ed-cadherin over here. So those are going to make it so those only stick to each other. And the E-cadherin up here makes it so this tube and this separates, right? Because they're not going to want to stick to them. So in the pre-migratory domain, BMP levels are at the highest. So before migration, BMP levels are the highest. With WNT at kind of an intermediate amount. And because of that, this increases the expression of SNAIL2, right? So if at the beginning state, BMP is the highest, WNT is at a normal middle level, and that supports SNAIL2 being regulated. SNAIL2 proteins then repress N-cadherin and E-cadherin in this domain. So because SNAIL2 is turned on, since this closure causes a lot of BMP and a medium amount of WNT, SNAIL comes in and says, all right, we're going to turn off all the anti-cadherin, right? And so when it does that, and it suppresses N and E cadherin, that allows these cells to not be sticky anymore, right? This causes that delamination. Let's see. So this causes this cadherin 6B here. That's upregulated only in the apical portion up here. And that functions to activate RHOA. Is that labeled on here? Oh, this shows RHOA. So it shows that it's the red part in these cells here. So this cadherin 6B activates RHOA and actomyosin contractile fibers. So in essence, cadherin 6B increases RHOA in these actomyosin stress fibers and allows further for them to pull off of the neural tube. So let's step back again. BMP is high and WNT is medium. We talk about these gradients all the time, right? So in this situation, SNAIL2 then is activated. A lot of SNAIL2 happens. This represses the cadherin and causes cadherin 6B to be expressed, which activates RHOA and these actomyosin stress fibers which allows them. So now we have no coherent, and we've activated ROA in these fibers to allow them to delaminate and be free, right, and go where they want to go. Okay. So during this process, there's kind of a fascinating and interesting thing here too. So what happens too is when these neural crest cells, after this expression has happened here, where we have the retraction and the delamination, so they've kind of pulled apart from the neural crest here, we get what's called contact inhibition. So you can see it right here. And this is this weird phenomenon where if these two cells run into each other, they'll go the opposite way. So it's like a dispersal. It's like putting oil in water or whatever. It's like causes things to disperse. So those, when they run into each other, when they experience this contact inhibition, they will stop, they'll turn, and they'll migrate away in opposite directions. So this kind of helps to create this equal diffusion into the two different, to like lateral sides, right? So here, there's also, so that's a general, this contact inhibition. But there's also, so that's like one method of migration. But there's also collective migration. So some populations of the neural crest cells are known to migrate collectively as a large group of cells. So this collective migration requires some amount of cell-to-cell adhesion, which we just talked about in the previous slide is usually not like the case, right? So if snail 2 comes in, we don't have cell-to-cell adhesion anymore. But this collective migration is mediated by a low level of N-cadherin, which are labeled as these blue receptors. So you can see here low level of N-cadherin. So these collectively migrating neural cells will also secrete an attractive signal. Is that labeled on here? Oh yeah, so that's the green goo, kind of C3A, to ensure that the neural crest cells continually kind of grow towards each other. So the pattern of migration by the group has kind of a collective direction due to the contact inhibition among the cells at the leading edge. So this causes them to kind of pull together, and then as the group kind of moves, that contact inhibition causes them to move as a group, as opposed, because they're stuck together, as opposed to one another. So you can see that like as they migrate up and start to contact this, they'll move in the direction of the contact. Okay, let's see. So there are two paths that this can occur. So this is kind of like a pulled apart. You can see we have the neural tube here with the lumen, and it's running to the caudal side and to the rostral side here. We have the sclerotome and the notochord. So we're going to talk about the dermomyotome and the sclerotome when we in the next chapter or two. But anyway, so you can see this is kind of, if you were looking, bottom up, you would see, we're used to seeing the notochord and the neural tube like this, and we'd have the neural crest here, and then the epidermis kind of wrapped around it. Well, there's two different paths that they can migrate through. So they can travel through the interior of the sclerotome. So this is where it's sneaking kind of along along the tube here and going out, or they can just kind of wrap around the outside of the tube this way. So the first is called the ventral pathway. So if they were to go, ventral is more interior, right? So kind of going from your back inside to migrate. And then the dorsolateral pathway is where they just migrate along the outside of the, near the epidermis, kind of on the outside layer to get to where they're going. So lastly, we'll wrap. What are we at? 25 minutes. Oh, this has been a long one. So we're wrapping up here. So there are differential gene expression that can cause or different mutations in genes that cause selective migration patterns. So in several animals, the random death of melanoblasts, so those neural crest cells that are going to become melanocytes eventually, so this is what provides the spotted pigmentation, like in these Dalmatians here. So these migrating melanoblasts induce the blood vessels to form in the inner ear, and without these vessels, the cochlea degenerates and the animal cannot hear in the ear. So if you've ever known anyone that has a Dalmatian, you'll know that they are often deaf, right? And so these genes have multiple functions, right? So in the migration, so because those cells aren't migrating to the inner ear and they're not migrating to the correct part of the epidermis, you have this co-occurrence of deafness and this pigmentation pattern. We also have something like these painted horses, which are thought to be heterozygous for endothelin receptor B, which causes this. You can also get piebaldness in babies, so this is kind of this unequal, where this unequal distribution of pigment, where pigment fails to form in the regions of the body, and this is a result of a mutation in a gene called KIT, K-I-T. This KIT protein is essential for the proliferation and migration of these neural crest cells. And then you have an example of mice that can also have this KIT mutation. And so this is really good. So for piebaldness, this is, you can see like maps right onto the belly, like just like, looks like the exact same pattern, interestingly, of the baby. But this is, this mouse that has this mutation in KIT as well is kind of a model for piebaldness study. So, all right, that's it for this. This was a bit of a longer one too. It's just kind of hard to break this one up into different topics. Thank you for watching. I hope you enjoyed the video, and I think we've got one more for this week, but I'll catch you in the next video.